All right. Well, I want to welcome everyone to the International Elephant Foundation's Conservation Chat. And we're really excited to have you join us today from all corners of the globe. Um, our discussion promises to be very insightful and fascinating. And I'm really looking forward to hearing our presenter. For those of you who don't know me, my name is Julie Bates. I'm the Donor Relations Manager for IEF. And some other staff members on the line with us include Deborah Olson, our Executive Director, Dr. Chase Ledoux, our Special Projects Manager, and Sarah Conley, our Director of Conservation. Before we dive into the presentation, I wanted to go over a couple of housekeeping notes to make sure we have a great presentation. As we're going through, we request that everyone please have their microphones off and their cameras off. This can help save the bandwidth for the presentation because we have people coming in from different parts of the world that may not have great internet access. Just so you know, this chat will be recorded. So it will be available after the chat on our website and through our social media channels. So you can revisit it later or send it out to people who you know couldn't make it today. And as the presenter is going through his dialogue, if you have questions, please, we encourage you to put those in the chat and then we'll get to those at the end of the presentation. And then when we're done, we'll invite you to maybe turn on your cameras and we will give everyone a round of applause. So without further ado, I think that has everything covered. I'm going to introduce you to Sarah Conley again, our Conservation Director, and she is going to be our moderator today and tell you all about our presenter. So take it away, Sarah.
Thank you, Julie. Good morning, everyone. We have a very exciting conservation chat today. I am super eager to share this presenter with you. We have someone who changed the face and landscape of elephant conservation. Dr. Alfred Roca is a genetics professor at the University of Illinois in Urbana-Champaign. He earned his doctorate at Harvard University, then conducted research at the Laboratory of Genomic Diversity before joining the University of Illinois Urbana-Champaign. He has published over 110 peer-reviewed scientific journal articles, including the first strong genetic evidence that African forest and savanna elephants comprise distinct species. This is a huge deal. His research interests include the population genetics of elephants and the genomic studies of wildlife, domesticated animals, and human health. So he's an expert in many things. We are very happy to have him, and please ask so many questions. He's a wealth of knowledge. Welcome, Dr. Roca.
Thanks, Sarah, and my thanks to the International Elephant Foundation for inviting me to give this talk, presenting some of my research. Some people I'd like to thank, Patricia Peknerova, Yasko Ishida, Steve O'Brien, Nick Georgiadis, many others. I'll show a slide at the end. On the left here, you see images of forest and savanna elephants. Savanna elephants are the world's largest terrestrial mammal. Elephants that live in Africa's tropical forests are smaller. They have thinner, straighter tusks, more rounded ears, a number of behavioral differences, and other differences in morphology. They were first described by Machi as being separate from the savanna elephant in, I believe, 1899 or 1900. However, the first detailed morphological study of Africa's elephants was not conducted until the 80s and 90s by primarily Cullen Groves and Peter Grubb. They measured the skulls of 275 African elephants. They could tell the age of the elephants by the molar wear patterns. Elephants have 6 molars that come out consecutively. This image, this slide was a little bit distorted in the original figure, so I'm just copying the original figure. But they were able to use a statistical discriminant function to completely separate not only males and female elephants, but also you can see that the Africana, which is the savanna species, could be completely distinguished based on morphology from the forest elephant. And so they proposed that 2 species be recognized. And you can see on the top right, there's a map showing the tropical forest range in Africa. That range is sort of approximately the range of the forest elephant. Our work begins with this gentleman, Dr. Nick Georgiadis. He, in the 1990s, went across Africa with a dart biopsy, which is, I think it's got a little piece of dental wire attached to it. It falls off the elephant when it hits it and just takes off a small bit of skin. That small piece of the outer skin is enough to get all the DNA you want for genomic studies. My work on this began in 1997 when I joined the laboratory of Dr. Stephen O'Brien. You can see here I've recently had reason to look back on my notes and so on. In 1998, we had alignments generated. On the right-hand side, you can see one of the 4 genes that we used in the initial study. That was in 1998. Bottom left, you see the results of 38 samples of African elephants. This was the first really good evidence that African savanna and forest elephants were distinctive from each other. You had different DNA values for the 2 elephants, a C in one case, a T in another case, A, C, G, and T are the 4 nucleotides of DNA. We ended up publishing in 2001 in the journal Science. We combined sequences from 4 different genes along with an outgroup, which was the Asian elephant. And we generated this phylogenetic tree. You can see next to the branches on the tree is the support level. This is a bootstrap statistical support. 100% support for all the Asian elephants forming a distinct category or clade. In the case of the savanna elephants, again, 79% support, 97% support rather, for all the African savanna elephants forming a distinct group or clade. And all of the forest elephants formed a distinct group or clade. Now, these numbers are a little bit lower than they would be because we found that there were some cases of hybrids. These had been reported previously since at least the 1950s. It was known that where the 2 species met, they hybridized. And you can see there's 2 populations, Garamba, which is right on the edge of the forest and savanna in the Democratic Republic of Congo. That sort of appears in both cases. So there's a little bit of forest and savanna in that region where the 2 habitats meet. And then we found out that the forest populations within Kenya, so these are forests in Kenya surrounded by savanna, those were just sort of pure savanna elephants. A couple of things I will point out here. One is that the horizontal distance is really the only thing that matters. And you can see there's a lot of diversity. The vertical lines are just connectors. They don't really mean anything. But the horizontal lines in the forest elephant, there's quite a bunch of them. This indicates that the forest elephants have very high genetic diversity. And this will, prove throughout, including now the genomics era, this has proven to be the case. Whereas the savanna elephant, there's very little diversity. In fact, all 4 genes were identical in sequence for elephants in southern Africa, as far as Namibia and in eastern Africa and in Cameroon. There were elephants that had very similar sequences or identical sequences. This was somewhat surprising because there are differences in the habitats that elephants live in. So the savanna elephants in Namibia live in desert regions, but they look just like other elephants. On the other hand, you have to consider that gene flow and migration within each species are really, there's nothing stopping them. So gene flow, very high in elephants, migration, very high in elephants, long distances involved. So there's nothing other than that forest savanna difference to keep the elephants from interacting with each other or to keep gene flow from going across the landscape. So I'm showing you multiple lines of evidence that supported the conclusion that these were genetically 2 different species. The monophyletic tree that I showed, where all of the forest elephants grouped together, all of the savanna elephants grouped together. This is an estimate of the genetic difference between the elephants based on full mitochondrial genomes. I'll talk a little bit about these towards the end. But this is my student, Adam Brandt. He looked at a few forest elephants, a few savanna elephants, compared them to Asian elephants or woolly mammoths. In this case, the estimate for the split between forest and savanna elephants was about 5.5 million years. So all the way back to around the time that Humans are believed to have split from chimps. Um, this estimate is a little bit dependent on some assumptions, but no matter what genetic markers we looked at or what we used, the difference between the forest and savanna elephants was about 80–90% as big as the difference between Asian elephants and woolly mammoths. So Asian elephants and mammoths are considered to be two separate genera. They split towards the start of the Pleistocene, so quite a long ways away. Um, During a time, and they actually, both mammoths, Asian elephants, forest and savanna elephants, they all evolved in Africa during a time, the Messinian salinity crisis, where the Mediterranean dried up and African habitats started to become much drier, and you got the development of grasslands. Another line of evidence is measures of genetic differentiation. This is FST. What it says is there's a lot of genetic diversity when you combine the species. How much of that diversity is due to differences within species or populations, and how much of that diversity can be accounted for by the difference between two groups, two species, two populations, etc. So between savanna, for example, and Asian elephants, about 99% of the difference is due to the difference between the two species. Neither Asian elephants nor savanna elephants are that diverse within species. So if you combine them, 99% of the genetic variation is due to that species difference. Well, what's interesting is, if you consider that anything over really 0.2, 0.25 is considered very great or great genetic differentiation. It's interesting that between the forest and savanna elephants, about 94% of the genetic diversity was accounted for by the difference between the two groups. So even though we had shown that forest elephants are highly diverse within the species, 94% of the genetic variation could be accounted for by the difference between the two groups, indicating, a great amount of genetic differentiation between them. This is a different study. These were genetic markers that we looked at. And what this software that we use do is it doesn't know whether they're forest or savanna elephants. What we say is split them up as best you can genetically into two groups, so that each group is sort of conforms to each other, so they all look like each other. So split them up in a way that makes sense genetically. And here, what you've seen is it's pretty much split up the groups into forest and savanna populations. I've colored the savanna component in orange and the forest component in green here. You see Garamba, that was that hybrid population. That shows some introgression, some mixture. Garamba is on the edge of the forest and savanna habitats. There's a little bit of gene flow from forest elephants in the savanna elephants of West Africa as well. But other than that, you see very little evidence for introgression, for admixture, hybridization between the two groups. There is a little bit of a signal, and I will mention that now that we have the genomics era, which is 3 billion base pair of DNA that we can compare, we can actually see a little bit of forest introgression into the savanna. But in almost all the populations, it's very low. It's much less than 1% forest introgression. When you consider that, the Eastern Congolian forest and the Western Congolian forest, those elephants are extremely similar to each other. There's very little differentiation across these great distances. The elephants in West Africa, East Africa, and Southern Africa, we find are fairly close to each other because of very high levels of migration, lots of gene flow. It's interesting that that doesn't homogenize the forest and savanna elephant populations. And then this is actually what convinced me that there are two distinctive species, is if you look at individual genetic variants, so DNA is A, C, G, and T, and this is just one small example. It's a single gene. It's a single nucleotide difference. You can see that a C and a C were present at these two positions in this gene. In the Asian elephants, this is probably ancestral. It's found in all three of the species. So this probably represents the ancestral sequence. And there's two mutated sequences. There's the 3, which is T and C. That occurs in the savanna elephants. About 96% of the savanna elephants have this variant. And then the middle variant in yellow, about 40% of the forest elephants had that variant. And then there's this hybrid zone in Garamba. All three variants are found there. But even though all variants are found there, forest and savanna variants, those variants are not going from the forest into the savanna, right? You don't see any of these yellow ones in the savanna elephants. Likewise, the variants that are present in the savanna, number 3, the one colored in brown here, it's not going into the forest. What this suggests is even though there's hybridization occurring, the hybrids are evolutionary dead ends. And subsequently, the data that we were looking at said that the hybrid males are just not very successful at reproducing. So they don't move the genes in either direction. So what keeps the species apart is they hybridize, but the hybrids are just not passing on their genes, and that keeps the two species separate. And then this is another gene where you can see we've looked at quite a number of individuals. At this point, for this gene, we've probably looked at 1,000 chromosome segments, 1,000 copies of this gene, and it's consistent. You almost never see introgression. In this case, it was complete separation between the forest elephants, shown here in green, and the savanna elephant sequences, shown here in blue. They're almost as distinctive as the Asian elephants sequences, shown in pink. So again, a lack of gene flow across the three populations. And so we used the biological species concept, which was promoted, among others, by Ernst Mayr, which says that species are groups of interbreeding natural populations that are reproductively isolated from other such groups. And Ernst Mayr asked the question, well, what about situations where two species hybridize? And he said that hybrid individuals or even hybrid zones do not violate the biological species concept if they do not disintegrate the genetic integrity of the species. So this is actually a very strict concept of species. And we went, even with this under the strictest concept, we thought that these elephants should be considered distinct species. And actually, after we published this, one of the very nice things about it was Dr. O'Brien, my mentor at the time. He got a letter from Ernst Mayr congratulating him and saying that Ernst Mayr had always thought they were two different species. And so we suggested in 2001 in our publication that the savanna elephant, Loxodon africana, and the forest elephant, Loxodon cyclotis, should be recognized as two distinctive species. And there's other differences I haven't talked about. They have different diets, different life history traits, etc. Unfortunately, the conservation agencies were not pleased with our conclusion. The IUCN African Elephant Specialist Group, which is a critical group involved in elephant conservation, decided to keep the status of a single species. They said that West Africa was a little bit uncertain. I will point out that West African elephants at the time made up about 2% of the entire African elephant population, so that really you're using 2% of the elephants to hold up the recognition of the species. Some populations of high conservation value may consist of interspecific hybrids. Well, there's hybrid zones between polar and brown bears, and there's hybrid zones between bobcat and lynx. You don't consider a hybrid zone to be of conservation value. Ligers, tigons, interspecies admixtures are generally not considered to be of conservation value. And the idea was that, well, we don't know, what all the populations are. In my book, that's not a very good reason for considering the two species to be just a single species and treating them as one. In 2008, they evaluated the status of the elephant. The elephant had been listed, I believe, as endangered. And they decided that it was no longer endangered, that the single species would be considered vulnerable, which is not as strong a conservation priority. And the reason they gave is, I'm citing it here verbatim, so you can read it. They said that populations in Eastern and Southern Africa are increasing quite a bit. Those in Central and West Africa, maybe they're not doing as well, but the increase in Eastern and Southern Africa, the number of elephants born in these populations will be larger than the total number of elephants in Central and West Africa. So the magnitude of the ongoing increase in Southern and Eastern Africa are likely to outweigh the magnitude of any declines in the other two regions. Now I've color-coded these. The important stuff is in yellow. The savanna populations that they're referring to are shown in orange, and the forest populations that they're referring to are shown in green. So the African Elephant Specialists Group is saying these elephants are not as endangered as we thought, and the reason they give is that you could completely wipe out the forest elephant in these two regions. And that they're not saying it's okay, but they're saying that the magnitude of the increase of savanna elephants would more than make up for the complete loss of the forest elephant. Sadly, in 2013, shortly after this, there was a study that said between 2002 and 2011, as the IUCN was choosing to sort of change the status from endangered to vulnerable, there was a 62% decline in the forest elephant population. A few years after that, Turcalo and colleagues found that there's a very slow intrinsic growth rate for Loxodon cyclotis for the forest elephant. Unfortunately, their age at first calving is older, and the interval between calves is longer. So it's much harder for the forest elephant populations to bounce back. In 2015, so 7 years after the IUCN decided that they were, vulnerable, and this is 14 years after I published the study, the Guardian published a piece where, we were saying, my lab and my colleagues were saying that we're condemning the forest elephant by not recognizing the two species. We said that the IUCN Red List should really list it as two separate species, and it would not be listed as vulnerable. It would probably be endangered or critically endangered. I compared what they were doing to saying that because the lion population is increasing, it would more than make up for the fact that tigers are going extinct, because tigers and lions split 3 million years ago, and elephants, the two species probably split even before that, maybe 5 or 6 million years ago. Georgiadis, my colleague, said that there's multiple lines of compelling evidence, zero evidence for just one species, compelling evidence for two species. Unfortunately, the conservation agencies, one official in the World Wildlife Fund that said that elephant conservation is already highly polarized in Africa, and recognizing the two species would further divide the nations on elephant conservation. I'm not sure why having two species that are more endangered than the single species would do that, but the article didn't say. Julian Blanc from the CITES program, said that they can interbreed and produce viable offspring. That's also true for lions and tigers and many other pairs of species. He also said that there's areas of hybridization, but also that there's known cases where savanna elephants live in the forest, such as Mount Kenya, and where forest elephants live in savannas. So there's parts of Africa where forest elephants may be living in a savanna habitat. To me, that's like saying mountain lions can't be a separate species because they don't all live in mountains. And again, I'll remind you at the same time, there were hundreds of thousands of forest elephants being killed for their ivory. Finally, in, I believe, 2021, I was contacted, I believe, by the New York Times and other agencies about the recognition finally by the IUCN. So this would have been about 20 years after our work was published. The African elephant species were recognized as separate. The savanna elephant went from vulnerable to endangered, and the forest elephant went from vulnerable, they skipped endangered, and it's now critically endangered, which is the last step before extinction. But I'm very glad that they finally recognized what to me was fairly obvious and very extremely strong evidence that there were two species. More recently, we've Illumina iConserve, which is It's a part of the company called Illumina, which is a sequencing company, DNA sequencing company. They have a mission to accelerate wildlife conservation, and they very kindly um, decided to help us sequence 400 elephants at 40x coverage. And the results I'm going to show are from the first half of these elephants. We've analyzed 249 genomes of elephants. 40x, for those who don't know, that's a very high coverage genome. So it makes sure that every part of the genome is covered with sequences. And so it's very reliable data. These were the same elephants that were collected in the 1990s by Nick Georgiadis, along with additional samples of elephants. And then we did, these are just some of the sort of the techniques that we used. And this is where the elephants were from. They were from across Southern Africa, Namibia, here in the Southwest, Southern and South Central Africa. Notice that there's gaps. I'll talk a little bit about that in our coverage. Elephant populations from East Africa. We now have samples from Uganda that were looked at. And Uganda is a place where the elephants were almost entirely killed off in the 80s and 90s. Two very isolated populations in Ethiopia and Eritrea, and then populations across Central West and West Africa, Sierra Leone, the only forest elephant we have from West Africa, and then Mali and Cameroon populations. And you can use the genome actually by scanning across the 3 billion base pair of the genome. You can estimate the lengths of fragments and how diverse fragments are, and combine those into estimating effective population size across time. This is a little bit different from census size because most male elephants won't reproduce. Male competition is very high among elephants. That lowers the effective population size to be lower than the census size. And then the number of years ago is approximation. Green shows the forest elephants. The other colors show the savanna elephants. What's interesting is when you look at the genomic level, there's a separation between forest elephants and savanna elephants in terms of effective population size. This is an indication that they were already separate, going back to where the different colors meet, which is probably about 3 million years ago. So consistent with the very ancient division of the species now being looked at on a genomic level. And then you can use a program that looks at principal components. What principal components do is they take this a genomic data set that's very large, and they say, how can you split this genomic data set in two where that split would account for the biggest difference? And then you erase that aspect of it, and then you say, what's the next biggest difference you can find in the remaining data? And it can do that serially. And what we found is the biggest difference between the two is it's called PC1 here, left to right. It actually separates the forest elephants on the right-hand side from the savanna elephants, which are on the left-hand side. You see a couple of hybrids. There are hundreds of individuals here, so only a few of these are hybrids. They kind of stick out, and the rest are here clustered. And then PC2, which is here on the y-axis, it's the next biggest difference you can find, and that actually separates the elephants found towards the west central part of the samples that we had versus the elephants found towards the east central samples. And that's what this separation is. Interestingly, when you go to the next level, PC3, that starts to divide up the savanna elephants geographically. And that's what this line here represents. And I will show you that in the next slide, where we only looked at savanna elephants. So now we're looking at the genomes of more than 100 savanna elephants from all different populations, and we're running the same analysis. And we're saying, separate out the populations by similarity to each other. And what you see is you get some clustering, but there's sort of a smooth distribution across the two principal components, with a very clear separation of elephants at the top here. Those are from Western Africa, savanna elephants. Eastern African elephants further along at one end, and then southern and southwestern elephants towards the bottom of this image. If you were to put a map of Africa here, this would actually resemble geographically the map of Africa. So this is genetics. The genetics of the elephants is following their geography. So you would expect western and west central savannas up to the north, then eastern savanna elephants with shown more on the right-hand side of the image, and then southern African elephants at the bottom here. You'll see some gaps here. Those gaps actually represent gaps in our data collection. They're probably not true genetic differences in African elephants. These are just, we don't have elephants from Chad or South Sudan, which would probably fit in one of the gaps, or elephants from southern Tanzania or Zambia that would probably fit in the other gaps. So it suggests that if we had complete sampling, it would be a continuous distribution. Now there is clustering of elephants, so all the elephants from one part of Africa, such as Mozambique, cluster together. All the Namibian elephants cluster together. This probably may represent a little bit of isolation. When things are isolated, they start to become related to each other. Animals become related to each other. So this may be a little bit of isolation, but the overall pattern is one of a continuous distinctiveness. So elephants that are further apart are a little more distinctive. Given that elephants migrate very long distances, they had continuous range. This is called an isolation by distance pattern. These colors do not represent necessarily conservation units because the elephants are not that distinctive. They're a little bit distinctive based on geography, but they're not sort of comprising separate species or subspecies, is what I'm getting from this chart. This is looking at admixture. It's a different way of presenting the data. And the one I point to is the one on top where the number of assumed partitions is 2. And that corresponds to the forest and savanna elephants. And you can see that the forest elephant populations are very distinctive from the savanna elephant populations. The regions of known hybridization, such as Garamba, you can clearly see the hybrid introgression. We've now got Uganda elephants where in the 70s and 80s, like I mentioned, 90% of the elephants there were killed off. Unrelated herds formed, forest and savanna elephants were mixed together, and you start to see that in Uganda. But overall, very little introgression across evident, even at the whole genome level in the savanna elephants. As I said before, we do detect a little bit using other methods that are more sensitive. We do detect a little bit of forest elephants introgression into the savanna elephants. You can force this program to separate out the populations even further. And you see savanna elephants from West Africa are a little bit distinctive. Forest elephants from the eastern part of the Congolian forest are a little bit distinctive from the ones that are in the western part of the Congolian forest. Not as deep a difference, obviously, as the two species, but you can detect that with the genomic data. This is just the savanna elephants, and you start to see, again, the West African elephants are distinctive. Namibian elephants can be distinctive. You start to get individual populations that you can actually identify using this method. Again, I would emphasize this is not subspecies. This is not, these populations can be distinguished using a very sensitive genomics method, but I would not call these sort of conservation units or subspecies. Although for sure you wouldn't want to lose all the West African savanna elephants, for example, or all of the Namibian elephants either. One of the things we looked at, and I mentioned in the first few slides, that forest elephants just seem to have a lot more genetic diversity than savanna elephants. Well, you can go across the genome and look for sites where the two copies of the gene are different. Elephants, like all mammals, they get one copy of a gene from the mother, one copy of a gene from the father. So you can go across the genome, look at genes and non-genic elements and say, how often do you see differences between them? How often were the mother and the father different? And that's an indication of how diverse genetically the population is. And what I'll note here is that the forest elephant populations are on the left. they're much higher in diversity. They have more than 3 times the heterozygosity, the genetic diversity of the savanna populations, which are much lower. So that's a big difference between them. The second thing is within the populations, they tend to be consistent. So almost all the savanna elephant populations have similar levels of heterozygosity. Again, an indication of the genetic connectivity that occurs. So again, remarkable that despite the genetic connectivity within the species, you get very little evidence of hybridization between the species. And then a couple of exceptions. One is you get really unusually high diversity in some populations. This is one of these is the population in Uganda. Another one is the population in Garamba. These are places where the two species have actually hybridized. And so some of these individuals have different hybrid ancestors, and that will tend to increase the genetic diversity. You can imagine if you're getting genetic material from both species that this would affect and increase the overall level of genetic diversity. And then some populations are just a little bit lower than is normal for their species. So on the far left, Sierra Leone seems lower than the other forest elephants. This is an isolated population. This is a population that's small and isolated. When that happens, you lose genetic diversity because the population is too small to maintain its genetic diversity. So that could be one reason for that. Populations in Ethiopia and Eritrea, again, those are isolated populations. Eritrea has fewer than 200 elephants for a very long time, for many generations. Isolated small populations lose genetic diversity, and that is going to be a threat to the elephants. And then on the far right, you see the elephants in Namibia have a slightly lower genetic diversity. The Namib Desert in Southwest Africa has expanded and contracted across geological time. What this low diversity reflects probably is the last time the desert contracted, elephants moved into the habitat, but only a limited number of elephants moved in, so they didn't have the full genetic diversity if a larger number of elephants had moved in. So this is probably a founder effect. So the elephant genomes are actually reflecting the effects of both natural and anthropogenic human-caused effects, especially in terms of isolation and loss of genetic diversity and inbreeding, actually. So this figure I'm showing now shows the level of inbreeding across the elephants. So inbreeding genetically means that mother and father have a common ancestor. So a great-great-grandparent, great-great-grandfather may have been also the great-great-grandfather of the cow may also have been the great-great-grandfather of the bull. And so part of their genomes will be identical coming from either parent because those two parents had a common ancestor. That's what inbreeding means in terms of genomics. And you can measure that across the genomes because they will show up as these flat desert regions where there's just no genomic variation in a part of the genome. And the length of that tells you how old these inbreeding events are, how distant the relatives were. And you can see that on the right-hand side are the savanna elephants, left-hand side are the forest elephants, much, much higher levels of inbreeding for the savanna elephants. For two reasons. On the left-hand side, the forest elephant, as we said, is much more genetically diverse. That suggests that a larger proportion of the elephants are mating. My guess is, this is not well established in terms of fieldwork, that the forest elephant males, more of them are successful. The males are maybe not as competitive. I'm not sure, but that's what this pattern suggests to me. And then in the case of the savanna elephants, males are very well characterized as being highly competitive. Older males, larger males tend to be successful at mating. Younger bulls are much less successful. So many males will not contribute genetically at all. So that means that the populations tend to have the same male ancestors, and that's what's causing the genomic signal of inbreeding. The other thing is that the populations tend to be very similar. The orange color at the bottom is actually very distant or very old inbreeding that's occurring. This is probably a natural pattern. And then above that are much more recent events or more intensive inbreeding. that's probably more anthropogenic. That's being caused by the separation of populations, the isolation of populations, limited elephant numbers, and that tends to increase inbreeding. And you see that in one forest population, that's the Sierra Leone elephant, because it's an isolated population. There's very few elephants left in West Africa. That is now being reflected in their genomes. Or actually, this sample was from an elephant, I believe it was born around in the early 60s. So it's probably gotten worse now. Luckily, I will say that for the forest elephants, we looked at their genomes to see like how many potential mutations do they have that might be expressed if inbreeding increases. And it wasn't as bad as we feared. So one bit of good news for the forest elephants as their habitat becomes more isolated and their numbers become lower. For the savanna elephants, you see higher levels of inbreeding overall. The more recent, more problematic inbreeding is seen in the middle part of this figure, where the elephants in West Africa, small populations, isolated populations, they're starting to show signs of that in their genomes. Elephants in Eritrea and Ethiopia, the same thing. And then to some degree, you see that in Namibian elephants on the far right. Other than that, the populations don't look too bad relative to the populations that are known to have been isolated for a very long time with small numbers. And then the final thing I'll talk about before sort of summarizing and taking questions is that we talk about the elephant genome. There's actually a second genome. So when we talk about genome projects and genomes, we're talking about 3 billion base pair that sit in the chromosomes, and that's what we think of as a genome. But there is a separate genome in the cell, and that's in the mitochondria of the cell. It's the energy powerhouse. It's only 13 genes, whereas the nuclear genome has 20,000. It's only 16,000 base pair, whereas the nuclear genome has 3 billion. But what's interesting is this separate genome, it's not passed on by the male at all. And that becomes very relevant for elephants because only females can pass on the mitochondria. And elephant herds, there's no female movement between herds. The gene flow that occurs across the herds is male mediated. And that includes not just the Y chromosome, which obviously is passed on by bulls to their sons, but all of the autosomal genes, all of the genes that are present in 2 copies, they're all passed on across the landscape by the males. Obviously, the mothers are passing them on to their offspring, but only the males are moving these genetic variants across herds. Even the X chromosome, which is found in 2 copies in females and 1 copy in males, the females don't move across the herds, and so the X chromosomes are moved by males. So everything is moved by male-mediated gene flow. But what about the mitochondria? Because it's going to be potentially very different pattern, a very unrepresentative pattern, because it's the only marker that cannot be passed on. Lots of markers are passed on by both males and females, but across the landscape, the mitochondria stays put. And as you know, female elephants or elephant herds are headed by matriarchs. Female elephants, calves stay with the herd. Males are kicked out of the herd at a certain age, and they move a genetic variation across the landscape. Well, you might expect mitochondrial DNA to be very different, and I don't want to go into too many details. It's extremely different. It's extremely variable. A lot of the variability, because the herds don't wander as far as males do, and they don't exchange females with other herds. You get very country-specific mitochondrial sequences. These might be good, and we have suggested that they are good for identifying the source of ivory. The one thing to keep in mind genetically is they don't take away from the 2 species concept. You get these very, very odd, very ancient separation of mitochondrial DNA. In fact, you can have 2 herds living in the same locality. Those herds are genetically indistinguishable. They look in every way like each other, but you look at their mitochondrial DNA, and their mitochondrial DNA are separated by millions of years of evolution, because there's never, the females are not exchanged between the herds, and that's only picked up by this one marker. And so you get this a very strange pattern that you can use to infer events in elephant evolutionary history. But the thing to keep in mind is that mitochondrial DNA does not reflect the rest of the genome. You can see these patterns as well. You can see the nuclear patterns in the orange and green on the right. They clearly follow the two species with a small number of hybrids, but they follow the two species pattern in orange and green, whereas the mitochondrial DNA is sort of all over the place, extremely diverse because of the specific inheritance pattern of that one single marker. Anyway, I will summarize here, and then I'll take questions. The support for the two species, morphological distinctions, which I showed a little bit, different life histories, fixed nucleotide site differences, one nucleotide, A, C, G, or T, present in the forest elephants, not present in the savanna elephants. Of the genetic variation, and remember, the forest elephant is extremely diverse, but 94% of the genetic variation was between the forest and savanna elephants. Despite that diversity in the forest elephant, only 6% of the genetic variation is due to differences within groups. That tells you how distinctive, how different the forest and savanna elephants are. In a tree, the forest elephants were all in one clade. Savanna elephants were all in a different clade or group. Very large genetic distance, 2.5 million years. Probably older than that. I would guess 4, 5, 6 million years. At multiple loci and multiple genes. Common genetic variants are restricted only to forest elephants or only to savanna elephants. Very few exceptions. There is hybridization, but the mitochondrial DNA, those female only DNA patterns suggest the isolation mechanism, which is that the male hybrids are just not reproducing. That's what we think, or are reproducing very little. And there's very little nuclear gene flow across the forest savanna elephant boundary, very likely due to the lack of reproductive success among the male hybrids. Anyway, that's the evidence we found. These are the folks. I thanked a few of them. This is the complete list involved in various aspects of the study, and I'll be happy to take questions. Thank you.
Thank you. That was impressive, and I absolutely think you proved your case beyond a shadow of a doubt. But we have lots of people asking questions. Why don't we start with, as a scientist, how do you view your role in conservation? I know you worked a lot to convince policymakers to take your evidence into consideration when doing conservation planning. So maybe talk about that a little bit.
Yeah, my role is to provide evidence, to provide data, to provide scientific conclusions. Can I be an advocate to some degree? And I think, I am, but that's separate from the scientific conclusions. So, I've had cases where folks tell me like they want there to be more species of certain animals because that'll increase their conservation, or more subspecies identified, or fewer for whatever reason. But I think that determination is separate from, what needs to be done for their conservation. What I would do different, if I had to do it over again, I would have just gone to every IUCN meeting, and I would have gone to every CITES meetings and presented this data, because there were easily a dozen scientific articles published consistently showing the same thing that I've shown, and I didn't go through all the articles or all the evidence. I should have taken that, because I don't think it's, there is a sort of very technical, very population genetics oriented aspect to it. Like, you have to know what the difference is between a microsatellite and a SNP, or Bayesian versus maximum likelihood analysis. But for showing what the patterns are, that's not really necessary. And I would have just, if I had to do it over again, I would have started going to every CITES meeting and every IUCN meeting and showing this data, because I think the data were pretty compelling.
Absolutely. I remember many years ago you presented at one of the IEF symposia, and I thought your data was extremely compelling at that point, and the official recognition hadn't happened.
No, it took a very long time. There was one point, this must have been a dozen years ago, when I got an email, and it was from the IUCN. They were taking a poll about what people thought, whether there were two species. And I wrote to the director, and I said, I've heard of a lot of species concepts. Like I talked about the biological species, there's a phylogenetic species concept, there's other species concepts. I said, I've never heard of a polling species concepts where you determine whether things are two species by taking a poll of people. So I just refused to fill out the poll. It's unfortunate, and I think the evidence was there. Um, why was it ignored? I don't know. It's probably considerations that were not related to the actual scientific evidence.
Sure. Uh, okay. Is there an idea as to what point, how many years ago, um, we can estimate that forest elephants became their own unique species? Like when did they break off?
You have to be careful about speaking about species that are extinct or fossil forms. So, for example, you can say humans and chimps are believed to have split about the same time, 4 or 5 million years ago. But there were a lot of intermediate species between the time of the split and modern humans. So modern humans appear, I think, a few 100,000 years ago, but there was Australopithecus. There were other species as well along the way. So you can have a change in species that probably happened in both, at least in the savanna lineage, where there were other species of loxodon in Africa. But the split between the lineage leading up to the forest elephant and the lineage leading up to the savanna elephant, my best guess would be about 5 million years ago, when Africa became drier, at the very end of the Miocene, I think, start of the Pliocene.
Interesting. Um, we have many guests on Conservation Chat, and one of our previous guests, Dr. Paul Ling, is here and asked about herpes viruses. Since herpes viruses co-evolve with their hosts, would you predict that forest elephants should harbor distinct but related herpes viruses to those found in savanna elephants, and the divergence would be reflective of the divergence between those species? And he goes on, therefore, might herpes virus genome sequence differences provide yet more evidence for species separation, not that you actually need more, or even reflect diversity within the species?
That's a good thought. I don't know that much about the herpes virus in elephants. I know it's fatal in some cases, and you don't keep elephants of different species together, in part for that reason. But um, whether the herpes virus would reflect that, I don't know. I guess it depends on how easy it is for them to cross the species barrier more recently. So I know some viruses, like SIV, crossed into the human populations to cause HIV only in the last 100 years. So I don't know what's the case for herpes virus. Certainly, it would be worth looking at. I don't know that anyone has looked at herpes virus in forest elephant, but I don't follow that so closely.
Okay. And he's got a follow up about hybrids. So while the hybrid elephants may be dead ends, is it possible that transmission of forest elephant specific strains could spread and be maintained in savanna populations? But I think you kind of talked about that. And is it possible that mixing or even cross species transmission of forest and savanna elephant herpes virus might impact the health of the hybrids?
That's a good point. Maybe it could. I don't know what is involved. When I originally wrote about it, what keeps them apart, I used the much larger size of the savanna elephant males. So fully grown, a savanna elephant male is twice the size of a forest elephant male. Larger male elephants are much more successful reproductively. So my thinking was the hybrids wouldn't be able to compete against the larger savanna elephant males. That would only hold true for a certain number of generations before the hybrids kind of, if it was only 1/16th forest elephant, it might be okay at reproducing. And the other thing it doesn't explain is why we don't see introgression of savanna elephant alleles into the forest elephant. So there are other things involved. Potentially, I guess, disease could be one of them, or it could be any number of issues. It would just be a wild guess if I came up with a reason. Could even be mitonuclear. I don't know.
No, you're doing great, and that is fascinating. We have a couple questions about the lack of genetic diversity in savanna elephants. First, do you think that's potentially due to the high rate of killing elephants for ivory in the early 1900s?
No, I don't. I think that's a natural pattern because even the very large populations that occur in some places, that hasn't changed. I don't see it. I think the genetic diversity is slightly lower in populations that have been isolated for a very long time due to human activity and have been small. But overall, it's pretty much the same across Africa. I don't think it's that reason. I think it's probably due to the high rate of male-male competition in African elephants. And I wouldn't say that the genetic diversity is low. I would say the forest elephant genetic diversity is unusually high because so if you look at Asian elephants or historical mammoths living in the Pleistocene, they're closer to the savanna elephant than they are to the forest elephant.
Interesting. Okay. Then do you think that low genetic diversity in savanna elephants should be taken into account in future assessments of IUCN status or any sort of conservation focus?
Um, I think you have to take into account the fact that populations in, say, West Africa are somewhat distinctive from Namibian populations, and so they may deserve their own conservation plan for that reason, because the two populations are not the same genetically. Whether the level of genetic diversity, I say you have to take it into account in a small population. Maybe has a high degree of inbreeding, whether you want to translocate individuals, but probably not overall in the conservation plan. Obviously, you want to maintain connectivity. You want to maintain large populations because losing that would be bad for the elephants genetically, especially in terms of things like their immune system genes, where diversity and variability is good for recognizing lots of pathogens.
Excellent. We have another EHV expert in the chat mentioning that recent research has shown that herpes viruses evolved with their hosts and that fatal disease isn't a result of mixing elephant species, but that doesn't mean that it doesn't bolster, if you find a herpes virus that co-evolved with forest elephants, the genetic divergence between the two species.
Okay, yeah. The one thing I will mention though is I think it was in the 1980s in the Chester Zoo, there was a female, I think, Asian elephant kept with a savanna male, and they actually produced a calf. They produced an interspecies calf. So it is possible for even across the two genera of elephants, Elephas, Asian elephant, and Loxodon, the African elephant, for the two species to hybridize. Unfortunately, the calf, I think it was named Madi, it died of a very common childhood calf infection among elephants.
Crazy. But then again, that goes back to that Liger analogy you had. So we're coming up on the hour. So maybe let's wrap up talking about, with all these modern genetic and genomic tools that you're using, what new questions about elephant conservation can we answer today that maybe we couldn't answer 10 or 20 years ago?
I'd say that genomics is showing the one thing I didn't emphasize is we have looked now, we were afraid for the forest elephants, or I may have mentioned this very briefly. One of the things we were afraid of is there's a tremendous amount of genetic diversity in the forest elephant, which is generally a good thing. But sometimes that means that they harbor recessive, so they're not expressed mutations that can be harmful. So when a population goes from a very large size very quickly to a very small size, like for an example is the case of the Florida panther, where it used to be connected with panthers or pumas across North America. It's a subspecies of the puma. And it became a very small isolated population. It started to develop things like heart valve defects and cryptorchidism. Just these genetic mutations started to be expressed in the population. They were probably there before, but the population was large enough that they were very rarely expressed. When they're present in a small population, you get inbreeding. That's when you get the high levels of expression. We looked at the forest elephant to see how common these sorts of mutations might be. And the one bit of good news is we didn't find that they were, we found that they were about the same level in forest and savanna elephants, which suggests that, genetically, it may not be as terrible for them as we feared. The other thing to keep in mind is that this low level inbreeding that's occurred in the savanna elephants, that can actually purge deleterious mutations. So over time, mutations that are recessive, you need two copies. If there's a little bit of inbreeding, they will show up and affect the elephants in a negative manner at the individual level. But because those elephants may not reproduce as much because of the genetic disease, that those variants tend to be removed from the population. So that both are sort of bits of good news revealed by the genomics data.
That's fascinating and a great way to wrap this up. I think little bits of good news like that leave people with hope and also pique interest for future studies. Thank you for this impactful work and for all you're doing. I know everyone is so excited to learn about this, and people are already asking when they can watch the replay and study this more. So let me turn it over to Julie.
Okay, great. I'd like to invite everyone to turn your cameras on and let's give Dr. Roca a big round of applause. And thank you so much for this wonderful talk today.